Hello, everyone. I am Jong-Hyuk Lee, and I'm an assistant professor in the Department of Biomedical Sciences at Mercer University School of Medicine. My talk today would be discussing my research about epigenetic histone modification and poly-ADP ribosylation dynamics during human aging. I would like to start with introducing Mercer University, where I do my research. Mercer University is located in Georgia, USA, and was first founded in 1833, named after respected Georgia Baptist leader Jesse Mercer. Main campus is located in the city of Macon, and other remote campuses are located in Atlanta, Columbus, and Savannah. My lab is in Savannah Medical Education and Research Building with very nice view of many palm trees. Savannah is also called the beauty of the South. This coastal city is the oldest in Georgia with beautiful large oak trees draped in Spanish moss shading historic squares and streets. We have beautiful beaches nearby, and we also have trendy boutiques and restaurants in downtown where shops on the famed River Street serve up candy, cocktails, and fun. So let's talk about research. We grew old. No matter what we do, it is inevitable, at least until now. So scientists worked really hard and found these 9 characteristics, also known as hallmarks of aging, to better understand the true nature of aging process. I'm the one who is also interested in one of these hallmarks. I'm particularly interested in epigenetic alterations, especially with histone modification changes during aging. This is called the heterochromatin loss model of aging. Heterochromatin refers to the dense part of chromatin, and it is known that once densely packed heterochromatin structure gradually lose their integrity and decondensates by aging. Epigenetics is the study of alterations in gene expression that does not involve changes to the underlying DNA sequences. For example, every cell in our body have exactly the same DNA. However, our skin cells are quite different from our intestinal epithelial cells. This is granted by epigenetic modification-driven terminal differentiation. Currently, DNA methylation, histone modification, and non-coding RNAs are the best characterized epigenetic modifications. I'm particularly interested in histone modification. Human DNA stretches about almost 2 meters in its total length, and it must fit into a single nucleus, which is only 6 micrometers. Higher-order eukaryotes have evolved to solve this by generating histone proteins. Histones wrap DNA and makes the beads on a string structure to form even higher-order nuclear scaffold. One interesting characteristic of these histones is that it has particular N-terminal tail structure, and these histone tails protrude into the nuclear plasm and are prone to diverse post-translational modifications. These modifications change the overall electrostatic charge of the histone proteins, thereby affecting the interaction strength with inbound DNA, ultimately regulating the density of the chromatin. So, when the chromatin structure is open to euchromatin, it exposes more naked DNA and grants access to transcription factors and coactivators to turn on the transcription. On the other hand, when chromatin is closed to form heterochromatin, these factors cannot access to the DNA and transcription is impeded. There are particular histone tail modifications that represent the overall status of chromatin. For example, H3K9 trimethylation. The trimethylation on 9th lysine residue on core histone H3 is abundant on condensed chromatin and represents heterochromatin structure. And this is achieved through modifiers that methylate histone tails, such as sub39H1 and setDB1, which are called histomethyltransferases. One of the earliest models proposing the involvement of epigenetics in aging was the heterochromatin loss model of aging. This model suggests that heterochromatin domains established early in embryogenesis are broken down during aging, and this causes global nuclear architecture changes and the repression of silenced genes and aberrant gene expression patterns. After the initial proposal, many follow-up studies have reported that the heterochromatin representing histomodifications such as H3K9, H3K27, and H4K20 trimethylations are decreasing by cellular and organismal aging. In addition, all of the core histone expressions are also found decreasing by aging. So taken together, all of these changes are pointing towards the same direction, that chromatin density decreases by aging. Nowadays, it is well established that the decreased heterochromatin exposes more unprotected DNAs to make it vulnerable to environmental stressors, such as ionizing radiations and reactive oxygen species. Also, heterochromatin loss results in failure of transcription repression of mutated DNA, transposable elements, and satellite repeats, resulting in reduced transcription precision and increased genomic instability, which leads to aging. In addition to chronological aging, genome-wide heterochromatin loss has been reported in some models of premature aging diseases. Premature aging diseases, which is also called progeroid syndromes, display signs and features of normal aging in early life, ultimately leading to premature death. Although progeroid syndromes do not perfectly mimic chronological aging, but it can be an excellent model to study characteristics of normal aging, especially when it comes to common features. Heterochromatin loss has been also reported in Hutchinson-Gilford progeria and Werner syndromes, although the precise mechanisms are still unclear. We were interested in studying another type of premature aging disease, Cockayne syndrome, and I was wondering if the heterochromatin loss also takes place in Cockayne syndrome model. Cockayne syndrome, also called CS, is a rare autosomal recessive accelerated aging disorder characterized by early onset neurodegeneration, vision, and hearing loss. CS patients shows abnormally increased mitochondrial respiration, accumulation of damaged mitochondria, and impaired mitophagy. The cause of disease mostly comes from genetic mutations in CSA or CSB gene, which are members of transcription-coupled nucleotide excision repair. One of the molecular characteristics of CS is that it has accumulated intracellular poly-ADP ribosylation. Poly-ADP ribosyl polymerase, PARP, is recruited to the site of DNA damage and generates PAR chains, poly-ADP ribosylation chains, using intracellular NAD as a substrate, and making these sites for DNA repair factor recruitment to facilitate DNA repair. However, without functional CSA and CSB, PARP1 may reside on DNA damage, and persistent PARylation causes intracellular NAD deprivation that leads to mitochondrial dysfunction and accelerated aging. So I raised some of these questions. Is the heterochromatin structure really disrupted in Cockayne syndrome? And if it does, will it have causal connection with the pathological features of CS? It is reported that heterochromatin loss or nucleosome decondensation leads to enlarged nuclei. I compared CS patient cells with their counter-control cells. Nuclear staining of both CSA and CSB deficient cells showed increasing nuclear size. So I checked if the CS cells are having altered histone modification, which may reflect their chromatin status. Western blots showed greatly reduced H3K9 trimethylation, which is heterochromatin marker in CSB-deficient cells. Also, CSB-deficient cells had much more phosphorylated H2AX compared to CSA-deficient cells. This suggests that CSB deficiency having more steady-state DNA damage than CSA part, which may reflect more severe clinical features of CSB than CSA. To evaluate the relationship between H3K9 trimethylation downregulation and CSB deficiency, I generated doxycycline-inducible CSA and CSB knockdowns in commonly used lab cell lines, HeLa and U2OS. As a result, CSB knockdown resulted in H3K9 trimethylation reduction in both HeLa and U2O cells, suggesting that decreased H3K9 trimethylation is a direct consequence of CSB deficiency. When we performed genome-wide H3K9 trimethylation chip sequencing, we found decreased H3K9 trimethylation around gene coding regions, as well as in constitutive heterochromatin regions in CSB-deficient cells. And when we performed genome-wide PAR sequencing, we found increasing PAR accumulation on gene coding regions, but not on constitutive heterochromatin regions. Our PAR peak enrichment calculation showed a marked increased PARylation at transcription start site, immediately downstream of transcription start site, and promoter regions in CSB deficient cells, all of which are proximity of gene coding region. I found decreased expression of H3K9 trimethyltransferase ZTB1 in CSB deficient cells, which I thought might be the cause of decreased H3K9 trimethylation in these cells. To test the effect of ZTB1 expression on heterochromatin in detail, I performed H3K9 trimethylation chip sequencing with CSB deficient cells that are induced to express ZTB1. As a result, when we induce ZTB1 expression in CSB patient cells, the H3K9 trimethylation significantly increased on gene coding regions. And with H3K9 trimethylation increasing by ZTB1 overexpression, hydrogen peroxide-induced PAR accumulation decreased on transcription start site regions of CSB deficient cells. It is reported that inhibiting PAR accumulation can rescue mitochondrial dysfunction in CSB deficient cells. So I tested further if PAR downregulation by ZTB1 mediated H3K9 trimethylation recovery can rescue mitochondrial dysfunction in CSB deficient cells. Mitochondrial oxygen consumption rate measured by seahorse extracellular flux analysis confirmed that CSB deficient cells having elevated basal and maximal respiration compared to control cells. Interestingly, ZTB1 induction rescued this into normal levels. These data shows that ZTB1-mediated heterochromatin recovery can suppress PAR accumulation around gene coding regions and rescue mitochondrial dysfunction in CSB deficient cells. Until now, I showed you how induced ZTB1 expression can rescue decreased H3K9 trimethylation and downregulate PAR accumulation and normalize mitochondrial dysfunction in CSB deficient cells. I wanted to further evaluate this model in relation to chronological aging. CSB and ZTB1 protein levels in fibroblasts from healthy donors from age 20, 40, and 60 years old were analyzed by immunofluorescence staining. And fluorescence quantification analysis showed that both CSB and ZTB1 protein levels were significantly decreasing with aging. This suggests that ZTB1, H3K9 trimethylation, PAR, mitochondrial pathway model in CS patient cells can also be applied to chronological aging as well. Finally, I tried to evaluate the age-dependent CSB and ZTB1 expression further in mouse brain tissue extracts from mice of different ages. As you can see here, I found age-dependent PAR accumulation and decreased CSB and ZTB1 expression, along with decreasing H3K9 trimethylation. This once again indicates that the heterochromatin loss resulting from ZTB1 downregulation caused by CSB deficiency may happen during normal aging as well. To summarize these results, when there is functional CSB, there is sufficient amount of heterochromatin maintenance by ZTB1, which results in minimal DNA damage and PARylation and healthy mitochondria. On the contrary, in CS patient cells and during chronological aging, impaired CSB reduces ZTB1 expression, which could lead to reduced H3K9 trimethylation on gene coding regions, triggering accumulation of PARylation and mitochondrial dysfunction. So, with these set of experiments, now we have the answers to the questions. Is the heterochromatin structure disrupted in CS? Yes. We found CSB-deficient cells having decreased H3K9 trimethylation than in control cells. And does the decreased heterochromatin have causal connection with the pathological features of CS? We found heterochromatin restoration rescued oxidative stress-induced PARylation and recovering mitochondrial abnormalities in CSB-deficient cells. Until now, I showed that heterochromatin formation regulated PAR accumulation, and PAR accumulation played a critical role in mitochondrial dysfunction. This line of nuclear PAR signaling to transduction pathway to the mitochondria is known as nuclear to mitochondrial signaling. According to this model, upstream sensors such as PARP1 ATM senses the DNA damage, and throughout the cascade of signaling pathway, downstream effectors regulate mitochondrial biogenesis or cellular metabolism, which leads to cancer and aging. In addition to this nuclear to mitochondrial signaling, I hypothesized that there would be more direct role of PARP1 on mitochondria. Traditionally, the existence of mitochondrial PARP1 is somehow controversial, and the biological roles for mitochondrial PARP1-induced mitochondrial PARylation are still unclear. So I thought it would be interesting to investigate the direct connection between PARP1 and mitochondria. After intramitochondrial PARylation was first found in 1975, it has been consistently observed and studied. However, mitochondrial PARP1 existence was a matter of heated debate in the field until very recently somehow. This is taken from a review paper, and we can take a look what people think about mitochondrial PARP1. It says, as far as intramitochondrial PARylation, the field was largely neglected and has not been studied systematically. Moreover, whatever little work has been performed in the field of mitochondrial PARP, it yielded divergent results. Some investigators find evidence of PARP enzyme in mitochondria, while others did not. So we concluded that the reason why people are having debates about mitochondrial PARP1 existence is simply because it is not studied systematically. So we tried to solve this by studying it systematically. We performed mitochondrial purification from wild-type and PARP1 knockout cells and investigated its purity by Western blot. Our mitochondrial preps showed absence of tubulin, indicating free of cytosolic contamination. And our samples were also free of lamin A and H3, showing free of nuclear contamination as well. In this condition, we could find PARP1 existence in our purified mitochondria from wild-type cells. We then wanted to make sure if this is not coming from outer mitochondrial membrane contamination from PARP1. We stripped mitochondrial outer membrane with optimized proteinase K treatment. As you can see here, proteinase K treatment removed TOM20, which is an outer mitochondrial marker, and maintained HSP60, which is a matrix marker. In this condition, we could still find PARP1 in suggesting PARP1 localization in mitochondrial matrix. We once again performed some mitochondrial fractionation with stringent quality control to investigate PARP1 localization in outer, inner mitochondrial membrane and mitochondrial matrix. With these, we found some amount of PARP1 in inner mitochondrial membrane fraction, but most of the signals were from mitochondrial matrix. And we were able to generate PARylation map on mitochondrial genome using biochemical technique called ADP-Ribosylation chromatin affinity purification. PAR distribution showed dispersed enrichment over mitochondrial genome. It was a bit dense in D-loop region and scarce in ND2 region. The overall distribution also showed inducing pattern by NAD treatment and suppressed with PARP1 inhibitor treatment. This pattern was not found in purified mitochondria from PARP1 knockout cells. It is known that histone density is closely related to chromatin PARylation in the nucleus. We hypothesized it would be similar in mitochondrial system. Although mitochondrial nucleoid does not have any core histones, it does have something similar called TFAM, which stabilizes mitochondrial DNA by packaging it into mitochondrial nucleoid. We then performed TFAM chromatin immunoprecipitation and PAR sequencing on mitochondrial nucleoid. As a result, TFAM and PAR distribution on mitochondrial genome showed strikingly similar pattern. And when we plot TFAM chip and PAR sequencing signal intensity on 37 mitochondrial genes, it showed very nice positive correlation. With TFAM chip, we found increased TFAM occupancy on the loop region of the mitochondrial nucleoid with NAD treatment. TFAM is one of the core mitochondrial DNA transcription factor and is known to induce transcription when bound to mitochondrial D-loop region. So we hypothesized that NAD-induced TFAM recruitment to D-loop may affect mitochondrial DNA transcription. So we checked mitochondrial gene expression, and we found mitochondrial transcription is actually significantly inducing with NAD treatment in wild-type cells, but not very much in PARP1 knockout cells. Again, this suggests that NAD-induced TFAM recruitment to the D-loop region regulates mitochondrial transcription. With these sets of experiments, including the ones that I've showed you today, we confirmed that the existence of mitochondrial PARP1 and PARylation. And we found mitochondrial PARylation regulating mitochondrial DNA transcription by TFAM recruitment on the loop region of mitochondrial DNA. And lastly, I would like to show you one of my latest research projects. Today, I showed you how Cockayne syndrome patient cells have decreased heterochromatin that potentially affects mitochondrial dysfunction. My ongoing research goal is to find out if there is any heterochromatin alteration in other types of premature aging disorders, ataxia telangiectasia, and xeroderma pigmentosum, that could be related to their molecular characteristics of the disease. So when I looked into both ATM and XPA patient cells by Wissem plot, there was significantly increased PARylation compared to control cells. I hypothesized that these elevated PARylation might have been caused by increased DNA damage susceptibility due to heterochromatin loss. However, unlike my findings from CSB, I found significantly increased H3K9 trimethylation in these cells instead. I tried to find out if the decreased heterochromatin content, increased heterochromatin in the content in these cells resulted in altered DNA damage susceptibility by oxidative stress. In ATM patient cells, we could find that decreased DNA damage susceptibility by hydrogen peroxide treatment. This findings is in line with my earlier observation that ATM patient cells have increased heterochromatin, which may have protected DNA by providing physical barrier against oxidative damage. Again, these decreased oxidative damage-induced PARylation accumulation in ATM patient cells were also existed in XP patient cells as well. Coming back to the initial Western blot data where I found ATM and XP patient cells having higher basal PARylation, this was kind of opposite from what I was expecting. I hypothesized that the increased PAR accumulation could be due to decreased heterochromatin content, like in CSB cells, which may cause more oxidative DNA damage. However, the oxidative DNA damage susceptibility was actually lower in these cells, which are actually in line with increased heterochromatin content. So my next question is, why are the basal level of PARylation is increased in ATM and XP patient cells, despite having reduced DNA damage susceptibility by increased heterochromatin content? So that was the last slide for my research. And lastly, I would like to give you one simple take-home message. Everyone knows staying fit keeps you healthy. In addition to that, I would like you to remember that staying chromatin fit keeps you young. I hope you find me and my research interesting today. Thank you so much for your attention.